session.
Okay, we move on with the next speaker, so we continue to the two parts of the session together, which is Isabel Fletcher. Isabel is from the Wellcome Trust, and she will be talking about synergies between environmental degradation and climate variation.
Hi, good morning everyone, and thank you for being here and having me. So I'm just going to actually preface that I'm actually here with two hats on today. I'll be firstly talking with my more academic hat, talking about some of my research that I did as part of my PhD, and then also talking about some of our work that we're doing at the Wellcome Trust using data science to address the climate crisis. So let's dive straight in. So this is a project, as I said, that I did at the London School of Hygiene and Tropical Medicine with some collaborators in Venezuela and also UCL. And this project was focused on investigating the synergies between environmental degradation and climate variation. So to start off with, mosquito-borne diseases are highly sensitive to climate conditions. And this is because both the vector, so the mosquito, and also the pathogen, so the malaria parasite, is sensitive to temperature and rainfall. So we know a lot about these climate disease relationships. In addition to this, mosquito-borne diseases are also highly sensitive to land use change. For example, malaria is affected by deforestation in the Amazon, but this relationship is really complex. So often we see in agricultural frontier regions, which we can see here, deforestation creates really suitable habitats for breeding mosquitoes. However, over time, as agricultural frontier regions progress and develop, we see an increase in malaria incidence, but then this can also decline as a result of increased healthcare provisioning. So this relationship is complex. What we don't know, though, about climate variation and land use is how these factors compound together and interact to determine and influence mosquito-borne diseases such as malaria. So in this particular study, that's exactly what we wanted to explore. We wanted to explore how climate and land use interact to determine malaria transmission. So this study was focused on malaria resurgence in Venezuela. And Venezuela is a really interesting case study to study this because over the past 20 or so years, they've experienced a really dramatic increase in malaria transmission, which you can see from these graphs here. And this is interesting because historically, Venezuela had actually been quite good with their malaria elimination efforts. However, as a result of the political and humanitarian crisis that is still ongoing, there have been lapses in control efforts, surveillance, and access to healthcare, which has compounded and meant that mosquito-borne diseases such as malaria have been increasing at rapid rates. So in this study, we focused on a particular hotspot of malaria transmission. And this place was called Bolivar State, which is in the south of Venezuela, which is just shown here. And as we can see here, these maps show malaria incidence in this state. And as you can see, there are particular hotspots in the northeast of the state. Another reason why Bolivar State is an interesting case study is because this area is a hotspot of gold mining activity. And gold mining activity creates really suitable habitat for breeding mosquitoes. Here we can see these standing pools of water that are created through the creation of the mines. And then we also have an incredible amount of forest edge habitat here. So this is the intact forest. And this all creates really suitable conditions for malaria transmission combined with the fact that often these illegal mines, which are usually gold mines, have a lot of vulnerable migrants moving into the area, and often they sleep in informal settlements with little access to healthcare or medication. So, as I said, mining activity is quite rife in this state here. This map here just shows of all of the remotely sensed mining sites in the whole of Venezuela, about 95% of gold mines detected are actually in Bolivar State, which is where we conducted this study. And these images just show exactly what the habitat is like and the working conditions are like in those mines. So what did we do? So in order to investigate the synergies between both climate variation and land use, so the mining activity, we modelled the spatiotemporal variation in malaria incidence in Bolivar State in southern Venezuela. So we formulated a Bayesian hierarchical model for both falciparum and vivax malaria, which are the predominant malaria parasites in the region. And we did this using monthly case counts for a 20-year period. In our model, we also had a series of climate and land use covariates, and these were all from remotely sensed satellite images, which included temperature, precipitation. We also accounted for El Nino, and then we had land use variables, which included mining, which I showed you those spots on the map, and then we also accounted for forest loss and urbanization using annual land cover maps. Also, in order to account for unexplained heterogeneity, we also used spatio-temporal random effects in our model to account for neighbouring effects between regions, but then also the seasonal patterns of malaria transmission. We also, in order to investigate what the influence of mining was on malaria transmission, we compared these spatial effects with and without mining. And then finally, we also, to investigate the synergies, we looked at what the interaction between climate and mining was. So what did we find? So firstly, we found that mining was highly influential in determining malaria transmission. And we can see here from the estimates from the model that mining was associated with increased malaria transmission. We then also, as I said, compared the spatial random effects of a model with mining to one without mining. And we can see in panel C here that where we get a reduction in the random effects, so we get a reduction from blue to the red bars, that tells us that mining is accounting for the unexplained spatial variation in malaria. And here we go. We can see that in one of the parishes in Bolivar that has the highest number of mines, mining actually explained 51% of the spatial variation in malaria in this region. So what did we find about the interactions between climate and mining? Well, we found that high mining areas did have more malaria, first of all, which is the top panel. But then we also found that warmer temperatures actually exacerbated malaria transmission. But this relationship was only true for areas that had high amounts of mining activity here. So if we compare the curve here at about a peak of 26.5 degrees, which is where we get the highest amount of malaria transmission, we can compare that to low mining areas, and we see that there's not much relationship at all. So what did we find in conclusion? So as I said, mining activity was associated with malaria transmission hotspots in southern Venezuela. And then we also found that there were synergies between climate and land use on malaria transmission. In particular, the effect of temperature exacerbated malaria transmission, but this was only in mining areas. So what does this mean? This means that areas vulnerable to global heating and environmental degradation are at risk of mosquito-borne diseases such as malaria. So this is really important to consider in light of rapid ongoing global land use changes and also global heating. This also means that we need to target control efforts in the region to these really high-risk areas that are hotspots of transmission. So I'm going to switch now and talk a bit about what we're doing at the Wellcome Trust. We are using data science to address the climate crisis, and our team are working across Wellcome to address three main health challenges. The first being mental health, and then we've got climate health and also infectious disease as well. And so the work that I have been doing is working at the intersection between climate and health and infectious diseases. So what does data for science and health look like at Wellcome? Well, we kind of fund three broad topic areas. The first being digital technology. So that is the actual foundational infrastructure and technology and software that can be used, such as some of the modelling tools that Rachel was talking about this morning. Then we also look at digital policy and systems. So can we create governance frameworks that increase access and facilitate access to data and data science? And then finally, digital equity, which is becoming increasingly ever more important to consider. So how do we make sure that everyone that needs access to climate data actually has that access, and is it free and equitable? Great. So what are the key climate and health issues that we're addressing using data science? It was really difficult to pick just a couple, so please come and talk to me about the other ones, but these are just a few. So the first, as I mentioned, we fund a lot of data and digital infrastructure. This year, we committed about 10 million to fund the development of modelling tools for climate-sensitive infectious diseases, such as Rachel's ID Extremes project. And that has been all across the world. It's been global. We've been funding projects focused on dengue, malaria, but also waterborne diseases such as cholera. We're then also improving and standardising metrics for climate and health. So we've also awarded around 5 million to the Office for National Statistics in the UK for them to develop official statistics and metrics to actually help monitor and track the health impacts of climate change. And then finally, we're also funding trustworthy development of digital technology. So 3 million that we have awarded to the Harmonise project, which will responsibly collect ground truth data using drones to bias correct climate data and improve infectious disease modelling. And so with that, that's all I have for you today, but please feel free to ask any questions.
Thank you, Isabel. Are there any questions?
Yes. Hi, thank you very much for the presentation, for your talk. About your first project on mining and malaria, maybe I got it wrong, but your conclusion was that malaria or rates of malaria are positively correlated with mining, right? Maybe I misunderstand it, but do you think that there are omitted variables? Because it's not about the mining that is around there, right? This is about people who get together and maybe there is one tank of contaminated water or something. So it's not about the mining, but that many people on one spot.
Yeah, that's a really good point, actually. So I kind of see, when we looked at mining, there are kind of two areas. There's the ecological alterations that happen with the mining, which creates more suitable mosquito breeding habitats. But then also, as you said, you also need people there to transmit the malaria parasite. So it's kind of two factors there. It's the human side of vulnerable people actually going there and being there to transmit the disease. But then also you do get those ecological effects from the mining itself.
Okay, thank you.
Okay, so for the virtual audience, there is a question about if you have seen into any correlations between, let me see, population density and malaria incidence?
As in, did we account for it? Because we did account for it in our models. We accounted for population density.
And then you see that effect as well, so that there is.
Yes, so it was kind of accounted in the model, but we didn't explicitly look for that association because it most likely exists.
Okay, right. Okay, I have a question. So you are using your climatic models to predict the incidence of malaria, and I was wondering whether there are any actions by the governments of the regional governments to do something with this information?
Yeah, so that's quite a tricky one. So because of the context, it's a complex political situation, and a lot of the work that some of our Venezuelan collaborators have been doing is trying to bring this issue to the fore, and we haven't really directly been involved in government, and we're hoping that the more work we do to bring to light this issue and quantify it, that the government can do something. But as I said, the vast majority of the mines that we're looking at are illegal and informal, so they're very difficult to regulate. But we're hoping by highlighting this issue, something can be done.
Okay.
Thank you.
Thank you. Okay. Thanks again, Isabel. So we move on now with Lara Urban. Hello. And she will be talking about real-time genomics for One Health.
Great. Yeah, you can hear me. Yeah, thank you very much for the organizing team for inviting me, and thanks for all of you for being here after yesterday's celebration. So much appreciated. So I'm really excited to talk to you about real-time genomics for One Health today. And quickly before I start, I would like to say that I will present some of my work that I've done as a research fellow in New Zealand until recently funded by the Alexander von Humboldt Foundation. But I will also present some work from my newly founded Genomics for One Health laboratory at Helmholtz Munich, specifically Helmholtz AI, the Helmholtz Pioneer Campus, and the Technical University of Munich. So let's start by asking, what is One Health? Whereas I think the previous speakers have done an amazing job in introducing it already. So One Health is a concept that confirms that the health of humans and the health of our environment and the health of animals and plants and all living organisms is intrinsically, inextricably linked. Which makes a lot of sense. We have just heard a lot of examples about how this link is being established, but obviously the ongoing climate crisis is like strengthening this link further. If you think about, for example, the impact of air pollution on premature death in humans, or the impact of freshwater quality on access to clean water. And probably the best example is the ongoing COVID-19 pandemic, which has by now been accepted to be associated with the loss of biodiversity and wild habitats. So coming to the next word in my title, real-time genomics. So I'm really trying to use real-time genomics for monitoring and better understanding One Health. So with real-time genomics, I really mean we want to bring genomics in a rapid manner out into the field to the point of care, where we can actually measure like One Health-related issues. And this sort of real-time genomics is currently only possible using nanopore sequencing. So how nanopore sequencing works, how you see here, is we have nanopores embedded in a membrane, and there's an electrical current going through these nanopores, and we also have our DNA going through those pores. So this DNA is changing the electrical current that is going through the pores, which we then can then, using AI algorithms and powerful GPUs, base call in real time into our DNA sequence. So this has like a huge importance for point-of-care applications because it's really rapid. In the moment of sequencing, you actually get your data, but it also has other advantages. For example, you can measure any sort of DNA modifications using this approach that would change the electrical current, which people have been using for calling epigenetic modifications, but I will come back to that later. Another advantage is, as you can see here, the approach is completely portable. So this is the small nanopore Minion sequencer that you can see here, which we, in collaboration with Miles Benton, a fantastic researcher in Aotearoa, New Zealand, coupled with embedded AI machines provided here by NVIDIA, which are basically like powerful, powerful small computers with really powerful GPUs that we can then use to really, in real time, analyze our data at the point of care. So bringing these two together, how can we use real-time genomics to understand One Health better? And I would like to try to talk about 3 examples today from my own research. Firstly, in the realm of animal health, about biodiversity-related issues. That's research that I've conducted on critically endangered species in New Zealand. Next, environment monitoring. I will present an example of freshwater monitoring research back from my time in Cambridge, UK. And finally, how can we also assess the impact of environment on human health, for example, via assessing pollution. And here I will focus on air pollution, which in Europe remains the biggest environmental threat to human health. So let's start by the biodiversity-related research. So I went to New Zealand for my research fellowship to do research on the genomics of critically endangered species, to really try to leverage genomics to directly assist the conservation of species directly in collaboration with the conservationists here. And here are just some of the species that I've been working with. So New Zealand has many critically endangered species because of the loss of habitat, but also the introduction of invasive predators. And so this has led to like really small populations that are in addition really closely connected. So often we only have one population left. And the species are highly inbred with low genetic diversity. So this really means these species are like very vulnerable to any sort of catastrophic event, such as infectious diseases. So they could, directly be wiped out from one day to the other. And a very good example here is the kakapo parrot, which you see up here, which is a species that has just above 200 individuals left. And for the last breeding season, the newly hatched kakapo chicks have suffered from an aspergillosis outbreak. So a fungal infection that can actually kill the chicks. And you can imagine, with only 200 individuals left, really every single individual is important. So the problem is that we don't really know where these really pathogenic aspergillus strains are coming from. And because it's suspected that these infections occur because of the warmer winters and summers in New Zealand, we also assume that it's a disease that's going to be there to stay. So it's like of huge conservation importance to try and monitor the spread of this disease and to also just contribute to the health of each individual kakapo. So I took a helicopter to one of the kakapo islands. We went out at night and would swap the kakapo chicks, like you can see here, the oral cavities. We would really do nanopore sequencing in the field, and yes, this is a kakapo chick on the Minion. And then we could do the data analysis in the field using these embedded AI computes that I've already mentioned. And here, it's obviously really important that we can do everything in the field, because sending the samples off the island, where you only every second week have a helicopter, would take far too long, and the chick would probably be dead by the time that we could confirm aspergillosis. And we also wanted to train the conservationists, who you see up here in the upper right corner, so that they could actually continue using it in our absence, which is actually happening at the moment. So I show you some quick preliminary results of this like really ongoing project. So you just see a few of our chick samples and the relative aspergillus reads that we find in the saliva, and you see that the actually taking into account the range of the y-axis, the number of Aspergillus reeds we find is actually really low. But there also seems to be a baseline of Aspergillus in all of the saliva. Now, we haven't swapped any chicks that actually developed aspergillosis, meaning that we don't really know what we would expect as an Aspergillus load in the saliva of infected chicks. But I said, so this is like currently ongoing this year to, I mean, we don't really want Aspergillosis cases to happen, but we will have to confirm what our expectation is of an infected chick. But I quickly want to point you to another detail of this study. So because we also used adaptive sampling here, which is selective sequencing using nanopore sequencing. So this really, again, makes use of this real-time component of nanopore sequencing, because in the very moment that your DNA passes through the nanopores, you can use these powerful GPUs, and we were actually the first ones who did this in the field, according to my knowledge, base call the data, and then align it while the DNA is being sequenced to some sort of reference genome that you are interested in. And then either eject the read or continue sequencing, and like this, either enrich or deplete for any sort of digital sequencing information you're interested in. So we use this to enrich for Aspergillus, and I just show you some sequencing data here. So we provided the Aspergillus reference genome. In the left side, here you see the total amount of sequencing reads of a normal run and the comparative adaptive sampling run, and you see that the number of reads is more or less similar. However, if we look into the reads that actually map to the Aspergillus reference genome, we see that we can double our amount of targeted reads using this adaptive sampling approach. I mean, we can really use this to increase our sensitivity for detecting pathogens that we are interested in. So, but let's continue to the next project now, environmental monitoring. So I picked this freshwater monitoring project here because obviously access to clean water is very important, and it's changing because of the climate crisis, whereas the United Nations have actually made access to clean water one of their sustainable development goals for 2030. So we here used real-time genomics to monitor the water microbiome in the River Cam in Cambridge, and what we did is we went out and sampled the river upstream of the city, downstream of the city, and in the city, and would actually do a 16S RNA amplification to look for the bacterial diversity. We sequenced at different time points to also look into the temporal variability, and these are the number of reads we got. And here you just see an overview of like the general clusters of bacterial families. And of the samples that we got. These here are just basically the positive controls, these samples that are clustering here. But what we see nicely is that we have quite a few bacterial families that cluster together and that occur across the samples. So this is really the river core microbiome, which also contains like a lot of known freshwater species. But we were obviously interested in the bacteria that could actually impact human health. So what we did next is we looked into potentially pathogenic bacteria, and also, so the potential pathogenic, you have them here, and also into wastewater associated bacteria. And then again looked across our sampling sites here, upstream of the city and downstream of the city. And you really see that we have like a lot more potential human pathogens occurring downstream of the city, probably showing how the impact of humans onto the city actually reflects back by increasing like the pathogenicity of the microbiome. We also find like the wastewater bacteria coming up here at this location 8, which was nice to see because this was actually sampled just downstream of an outflow of a wastewater treatment plant, really confirming and that we can see like this residue from this treatment plant in our water samples. We also, however, found like a few pathogens, including a Leptospira, just downstream of this wastewater management plant. And that was interesting because that might mean that this direct human impact might increase like a pathogenicity in the water samples. And especially Leptospira is of huge concern in Cambridge and also in other rivers because it can affect rowers and other stakeholders of the river and can actually lead to death in the very worst case. So we followed up with this Leptospira and also with the other pathogens, but I'm showing you the Leptospira example right now, because obviously, like in a bacterial genus, you might have pathogenic strains, but you also have completely saprophytic strains that are expected to occur in the environment. So in order to increase the resolution for if this was pathogenic or saprophytic, what we found, we took all of the publicly available Leptospira data that we found, took the reads, combined them with our reads, did a multiple sequence alignment, and then created this phylogenetic tree, which basically here shows you like the outer circle here are the reads that we got from the databases, whereas the inner circles is again our reads coded according to the locations that we got the sample from. And what you can nicely see here is that these are the violet sequences here are the pathogenic ones, and the green ones are the saprophytic ones, that all of our samples, all of our reads cluster nicely with the saprophytic reads from the databases, whereas the pathogenic ones are like a separate cluster, probably confirming that we only have like saprophytic Leptospira in the river, which we then confirmed with some downstream analysis. So very briefly to say, obviously, I think for any sort of One Health related issue, also public outreach is really, really important. And this is something that we've really been pushing within this project, which we called PanSeq. And here I really want to mention Max and Andre, who have been co-leading this project with me. And I'm also happy to say that my lab's first master student, Anastasia, is actually continuing this work now on the metagenomic instead of on the amplicon sequencing level at the moment. So, but let's now come to my current work, which focuses a little bit more on the human health applications. So we and others have started imagining how we can actually use these real-time genomic approaches in the context of human health. And obviously, we can use it in the settings of hospitals, at places of high human density, such as airports and agricultural settings, and really all around the world, because it is so portable. So in the end, I think this real-time genomic approach can really help democratizing the benefits and opportunities of genomic research. And this is something that is really important in the One Health context, because this will obviously only work, like understanding One Health better and so on, if we do it in a global context. So we are working with several industrial partners to actually automate this entire process, to make it more available also like for other stakeholders outside of science and local communities, public health institutions, and so on. I quickly want to mention that we also work with the Microgem company, which is something that you computational folks might not be so interested in, but what is also obviously really important when you want to automate and simplify processes is to also automate the DNA extraction part. And here we are working on like making this a one-step process by just being able to put your DNA sample into a straw and then into another portable machine, and you directly get your clear DNA out. Thinking on a larger scale, we are also working with researchers in Munich to combine these like really detailed molecular data that we are producing and that we are ideally producing in an automated manner with like this large-scale remote sensing techniques, which is something that I think can be very powerful in combination in the future. So just to show you one very quick example, coming back to the air pollution part that I mentioned beforehand. So we started sampling air using the cyclonic air samplers here, and Tim, a PhD student in my lab, started establishing a protocol for actually using real-time genomics for understanding the air microbiome, so all sorts of bioaerosols, bacteria, viruses, fungi that are floating around, especially obviously focusing on pathogens. So as you remember, what I told you is like what we get from nanopore sequencing is this change in the electrical current, which is also called squiggle data, how they call it at Oxford Nanopore Technologies. And we can then base call this squiggle data into the actual DNA sequence, and then we can do like typical metagenomic research, which we've heard a lot about at this conference, doing like metagenome assemblies, taxonomic assignments, and also look into functional annotations by looking for biosynthetic gene clusters that tell us something about the pathogenicity, virulence, or also antimicrobial resistance of these bacteria. However, I've also started thinking about what else we can do with this sort of squiggle data that we get. I mentioned beforehand, we can also call epigenetic modifications from it. So we can basically try to look into differences of the squiggle data and think about what else can the squiggle data actually tell us. And I had this idea, and my other PhD student Harika is looking into this from October on, if we can actually, from the squiggle data, using AI, infer if a DNA came from a dead or a living bacterium. Because under the hypothesis that DNA from a dead organism has been floating around in the environment for a while, has been exposed to environmental damage due to UV, to temperature, and so on, and has therefore been degraded to, like, let's say on an atomic scale that we can then assess in the squiggle data. And this is obviously something really important, especially for air monitoring, because we are not interested in any like free-floating DNA that comes from dead organisms. We are interested in organisms that are virulent and potentially pathogenic. Yeah, but so this is ongoing work at the moment, and I'm also super excited that the lab's first postdoc, Albert, will actually use this bioaerosol monitoring plus viability studies in the future to monitor the spread of infectious disease in wild animal populations, such as bird populations, as you see here. And yeah, so with that, I will have to finish. I didn't actually talk a lot about my actual biodiversity research that Rachel mentioned at the beginning, and also not in detail about the human health-related research. So if you're interested in any of that, just find me in the break or just contact me down here. Thank you very much.
Thank you, Lara, so much. Are there any questions in the audience? Well, maybe you can come forward to the microphone, and you can come forward too, so she can start. Wait. We'll get her first. Okay.
Is it on? No. Thanks for Chantal. I wanted to ask about the kakapos. You've mentioned that you tried to sample to look for the infectious disease in chick samples, and you didn't have it there, so that you don't have a very good picture of how it looks when they are infected. But there were some infected birds, adult birds, in that season, right? So I was wondering if that could help you to say something about what to expect in chicks, or if you have that kind of data or whatever, if you can comment on that.
Yeah, absolutely. We have a kakapo expert here, it seems, who has followed the last breeding season. Yes, indeed, we had some adult birds as well this season that had fallen sick, but it was the same problem as with, we've also had a few infected chicks. The problem was just we did not swap them, because we could obviously not predict which individuals would get sick, and it was like, we randomly, basically, in a randomized manner, swapped individuals to understand, to predict if there was an outbreak in this individual. And we just, by chance, didn't swap any of the maybe 10 individuals that got it. But yeah, really good point. And yeah, I mean, we are looking into right now if we can detect any of the cases, yeah.
Thank you. Hi, Lara. Thanks for the talk. I have two questions. One is related with the Kakapo project. You have evidence that most of the chicks have the Aspergillus in the saliva, no? The pathogen. But I wonder, are you checking the genotype of the different chicks? Because some of them, yeah. Okay.
Yeah, that's a really good question. So actually, majority of my biodiversity work in New Zealand was actually looking into the genomics of the actual endangered species, right? So we did try to get the genomic data from the saliva from the chicks, but we just didn't get enough DNA out of them to sequence them on the nanopore. So we ended up using blood. So we just pinched like the kakapo chicks a little bit, got like really 1 microliter of blood that we needed. And because birds have nucleated blood cells, that was a lot easier to get DNA from. So we did that. It would have been amazing to combine like the sample and use it for both approaches, but at least in our case, like with this automated DNA extraction approach that we tried to use, we didn't get enough DNA from the saliva. Yeah.
Okay. And the other question is if you have some preliminary data about the squiggle data, because I guess if you have that bacteria there, the read lengths will be shorter in the sample with death.
The read length, yes.
Yes. The distribution will be different.
Absolutely. Yeah, so we actually created like some experimental data for this project, because obviously when we get samples from the environment, we wouldn't know which bacteria were dead and alive. So we had some E. coli cultures like nicely living and thriving and sequencing those, and then we let them die and sequence like at different time points after their death. And absolutely, you see that the read length is going down, considerable actually after a few weeks. However, this is not something we can use to actually annotate on a per read basis, right? Because the only thing we will see that we have a general different distribution, but we can't disentangle if we had like data from like living and dead bacteria mixed into which read was coming from a living or a dead bacterium, if that makes sense.
Yeah. Thank you.
Thank you.
We have one question from the virtual audience, and it's again about kakapo infection. So, whether you know where the Aspergillus infection originated from?
So that was also part of the project, actually, to look into like where this was coming from. So we swapped soil samples and so on to look into if we could find exactly the same sort of strain of Aspergillus in anywhere in the environment, but we didn't actually find it in any of the soil samples. So it's still a little bit curious, and some fear has been that maybe it has been brought in from humans onto the island, which would obviously be a bad side effect of the conservation efforts, but this is still something we have to confirm. So we haven't found the same strain anywhere in the environment, really just in the infected birds so far.
Okay, thank you, Lara. Again, we can thank her for the talk. And I have too many things. So we have the last talk now, which is Tilly Alcaina, right? Okay, she will be talking about climate-sensitive disease outbreaks.
Hi, everyone. It's a real pleasure to be here with you today. My name is Tilly Alcaina. I'm a PhD candidate at London School of Hygiene and Tropical Medicine, as well as the Senior Technical Advisor for Health and Climate at the Red Cross Climate Centre. And this morning, I'm going to be talking about some research on climate-sensitive disease outbreaks in the aftermath of extreme climatic events. So climate-sensitive infectious diseases are, for example, cholera, malaria, dengue, diarrheal diseases. They have a really high global burden and are of major public health concern. In this map here of Africa, between the years 2016 and 2018, a research group mapped how many epidemics had happened. And the darker the color is, the higher the number of epidemics. And what you can see is certain countries in which there is really, really dark blue are experiencing more than 20 epidemics in this 2-year time span. So that's really a lot. Climate-driven extreme climatic events, think flooding, think typhoons, think heat waves, think droughts. The increased risk of these in terms of their intensity and frequency risks a higher outbreak of climate-sensitive infectious diseases, which will require a greater public health response and a humanitarian response. And these are two systems which are already highly, highly overstretched. So it's really important to try and understand the link between climate-sensitive infectious diseases and extreme climate events. But to date, interestingly, there had been no collation of those links between extreme weather events and climate-sensitive infectious diseases. And also, there was no synthesis of the potential hydrometeorological triggers that might be driving these outbreaks. So what this research is really trying to do is identify those potential patterns in the climatic drivers of outbreaks, and also the consequences of the extreme climatic events, in order to help inform the types of public health actions and humanitarian response that makes the most sense in a given setting for the different types of disease outbreaks. And ideally, being able to identify the most effective range of public health actions could either prevent the potential outbreak from ever happening, which would be the ideal case, or minimize the impacts by taking that appropriate action and timely action also. In the humanitarian world, this is called anticipatory action, and the whole point of anticipatory action is really the proactive interventions or early actions that you can do between this very crucial window. If the clicker is, where is my, here, when you've received a warning, but before the event has come to pass, that you can trigger the release of money, conduct some early actions, and therefore safeguard the population before this has happened. And that's the really crucial bit about anticipatory action, that it operates in this window between the warning, but before the event has come to pass. And when we consider that in terms of infectious diseases, you would have a forecast of one of these potential hydrometeorological triggers. The trigger would come to pass. You enact any of the early actions or interventions that you seek to do in the hope to reduce the caseload and the number of cases here with a lower epidemic peak. Whereas if you hadn't done any of the actions and without any of this early warning, you might have a much, much bigger peak. So the aims of this review that I'm presenting now were to identify those potential hydrometeorological and climatic triggers that are important in driving outbreaks of specifically cholera, diarrheal diseases, malaria and dengue that I was looking at as part of this scoping review, and then assess the state of knowledge on how extreme climatic events interact with the underlying exposure and vulnerability factors. I conducted a PRISMA scoping review using and extracting peer-reviewed articles from PubMed, Embase, and Medline with search terms to do with the exposure of extreme weather, like I mentioned before, drought, floods, cyclones, typhoons, heat waves, and then the outcomes of interest, and those were the climate-sensitive infectious diseases of dengue, cholera, diarrhea, and malaria. And what you'll see here was that in trying to assess the sort of strength of evidence and also the agreement between it, I color-coded it. So along here, we have the evidence in terms of the amounts of papers that were published and the higher transparency, so fewer number of papers is higher transparency. More opacity, so darker colors, means that there was a higher number of papers. And then up the side is to do with the agreement, which was also color-coded. So if we sort of see green colors, that means that there was a high level of agreement, orange sort of medium agreement, and then blue there was low agreement, and gray was where it was such low evidence that I couldn't have an agreement qualifier, so that's in gray. And you'll see this later in some of the figures. So the results were that of the sort of nearly 2,000 titles and abstracts that were screened, in the end, I retrieved 90 studies that met the inclusion criteria. 16 of these were review articles, and 74 of these were studies which looked specifically at interactions between extreme weather events and climate-sensitive infectious diseases in a specific place. These were then split of these 74 studies because some of them looked at, say, two climate-sensitive infectious diseases or multiple different types of extreme weather events that could, for instance, affect malaria. I split these then into the specific extreme climate and the disease outbreak event. So think heavy rainfall cholera or heat waves dengue or flooding and diarrheal diseases. And that resulted then in 90 of these different events. My first set of findings was about those hydrometeorological triggers that I mentioned are really, really important when you're trying to set up an early warning system. And what I found there, you can see, again, down this side, it's the sort of agreement and amount of evidence assessment. But what I found was that a real common driver across cholera, diarrheal diseases, malaria, and dengue was extremely heavy, extended rainfall that usually resulted in flooding, which often leads to damage and destruction of infrastructure, displacement of people, and the disruption of critical services like water, sanitation, and healthcare facilities. That was really a very, very important driver across all these different diseases. High temperatures were also heavily linked with diarrheal diseases and dengue. But then, interestingly, with heat waves and dengue, it really depended on the maximum temperatures reached during the heat wave and also the duration of the heat wave. So how many days of heat wave was it? Was it 3 days or was it up to 2 weeks? And that sort of affected the potential for outbreaks of dengue at different time lags thereafter. Another thing that was very, very important, and Rachel mentioned this also in her presentation, was the sort of sequential events of extreme climatic events. So if there had been drought and very low rainfall, and this was then followed by excessive rainfall, that was then particularly important for outbreaks of cholera, diarrheal diseases, and dengue. And so when we're trying to compile all of this together and thinking about those early warning systems that are really important for humanitarian actors, we really need to think about the successive events. So what is happening before and after? Has there been a drought? Is there then rainfall? And then also think about the compound effects of what this means. What might a drought have meant on the health system prior to then a really big flood happening, as is the case that has happened in Pakistan just now. And then also the sort of temporal dynamics of the event itself. So when it came to cholera, a very abrupt type of rainfall was also linked with cholera outbreaks. Another one of the major findings, which was pretty interesting, was that there was higher evidence and higher agreement on the links between extreme climatic events and waterborne diseases than for vector-borne diseases. And we sort of discussed and interpreted this, that it might be because vector life cycles are extremely complex. As Isabel was mentioning, you not only have the sort of vector ecosystem and ecology that you're taking into account, but also the pathogen and then that interaction with human behavior. There's also a substantial number of Anopheles species for malaria transmission, which might account for why, in fact, malaria seems to have quite a lot less evidence and lower agreement. And then also there are a lot of non-linear impacts between extreme climatic events and vector-borne diseases, like I mentioned before, about the timing, the maximum temperatures reached and the duration of the heat wave, and what then that means in terms of the time lag of when you might see a vector-borne disease outbreak like dengue or malaria. And then finally, also, as Isabel mentioned, interventions are also really important. So the distribution of bed nets or larval spraying to get rid of mosquitoes, that might also mask what would normally have been an outbreak after one of these events. We also found that research was really not aligned to need. If you remember back to that very first map that I showed from a previous research group's study of outbreaks in Africa, there were a lot of countries in which 20 outbreaks have occurred within 2 years. But in these 90 studies that I was looking for, there is a huge underrepresentation of certain geographies in terms of the outbreaks that they have experienced historically, and then also their climate vulnerability. So for example, once again, lots of those countries in Central Africa in which there had been more than 20 or up to 20 different outbreaks, they were not really featured in these studies that are looking specifically at the links between extreme climatic events and outbreaks, even though it is highly likely that extreme weather events were very important in those locations because they're so climate vulnerable and likely interacted with the risk of outbreak. We also found a substantial lack of evidence on the links between extreme climatic events and then the underlying vulnerability and exposure factors. So beyond sort of things to do with water and sanitation infrastructure being damaged, there was really limited in-depth investigation of population demographics, of displacement, of migration, of underlying health status, disruption to health services, and also how human behaviors might have interacted and modified the relationship between extreme climate and outbreaks. Two really big knowledge gaps were how extreme climate events, and especially long-term droughts or extended heat waves, affected ecological systems, and that was one of the questions, I believe that happened before, in terms of, what happens when the ecology is changed in a place. What about if the species composition changes, or if agricultural productivity is changed? How does that also modify outbreak risk? And also how recurrent extreme climate events may increase vulnerability and compound outbreak risk. So if you have successive floods happening in a certain location, are the populations able and the public health authorities able to learn from that experience and thus mitigate the future risk? Or are the systems overwhelmed and thus resilience undermined? And so each time there is another event, people face heightened risk. A couple of limitations of the research and really a lot of the information out there. So this was largely to do with information from English and peer-reviewed literature, so it didn't take into account a lot of the grey literature, a lot of WHO reports, for example, or MSF reports. That was not included as part of this study because it was really trying to understand what is it there in the peer-reviewed literature that we can see in terms of those potential triggers and then the links. But one of the biggest limitations that we face is that across all data, everything is really an underestimate of the true global burden, and this is often because when public health systems experience an extreme climatic event or some kind of disaster, they often collapse, further reducing our ability to record the caseloads that are coming out, or also recording an outbreak from happening. Not all countries have really, really strong surveillance systems, and so a lot of outbreaks are missed. Most of the studies reported on those positive associations. So most studies had identified a link between the extreme climatic event and an outbreak of a climate-sensitive infectious disease, which is quite suggestive of reporting bias. The grey literature might have helped a little bit more to identify where extreme climatic events had not resulted in an outbreak, but there isn't a global database which does record all of that systematically. And where most of the studies did provide a reference group in terms of potentially the average number of caseloads in the previous year prior to an extreme weather event happening, they were not so clear and consistent on a number of other dynamic contextual factors, so those human behaviors or other factors impacting disease dynamics, socioeconomic factors, the level of population immunity, that could also have been used to understand outbreak risk. But those are really challenging variables to often include into a sort of data and model analysis. But the outlook from this is that with this identification of some of those potential hydrometeorological triggers, we can then use them to start building up early warning systems for climate-sensitive infectious diseases in this intention of getting ahead of the curve, of trying to mitigate those risks and using climate risk information as early as, for example, in this cholera risk model that was used in Yemen, as early as 4 weeks in advance to be able to identify when you might be entering a period of heightened risk, to try and get in those early actions, to try and set up the sort of roles and responsibilities that are needed between the different elements of, say, humanitarian action, but also public health authorities, to put in place to try and reduce the curve as much as possible. Some recommendations for future work is definitely to improve the geographic representation of studies. So that means looking much more into studies, or many more studies being conducted in low-income or lower middle-income countries, and especially fragile and conflict-affected countries. And ideally, the sort of targeting of research will be based on some kind of need, so whether that be vulnerability criteria, vulnerability towards climate change, or risk of outbreaks based on historic data. That will help to build that evidence base of climate disease dynamics that can be used to leverage and reduce that health equity gap between the information that we tend to know about high-income countries versus the information that we tend to know about low-income countries. Ideally, a lot more work will focus on understanding the causal chain between climate change, extreme climatic events, and disease outbreaks for both waterborne and vector-borne diseases. And also work to improve how data on extreme climate events is integrated with disease incidence data at different time lags, so in the short term, long term, and medium term. If we were able to have very robust and accurate, or pretty robust and accurate information coming even 6 months in advance, that could help inform, say, the logistics of MSF operations, or the logistics also of public health authorities. That would be really great in terms of getting the medication and supplies in. And then ideally develop reporting guidelines for future studies so that we have consistency on reference groups that are used, but also clear definitions of extreme climate events and outbreak thresholds, case definitions, and underlying vulnerability. Thank you.
Thank you. Okay, so do we have questions? Apparently, yes.
Yes, I would have more than a question, a suggestion on your conclusion and how this conclusion can get into concrete actions. I mean, to increase the coverage of studies and all that in other parts of the world. This is something. But there is one about reporting. And maybe you could have a very concrete action addressing the journals and actually sending them a letter, really putting together some words about recommendation of how reporting conditions should be and how they could put that into the instruction for authors. And this would be completely different to the other things that really are more beyond, outside your control or many people's control.
Cool. Yeah, great suggestion. That final bit about those reporting guidelines is sort of the hope to develop up a consensus among researchers of what it is that we will report on consistently in these kinds of studies. Thank you.
Hi. Thank you very much. That was very informative and very interesting as well. You touched a little bit on that, but I was wondering, how do you get that data? Because you mentioned that some studies don't include countries with high number of outbreaks. And I wonder, is that kind of a global reporting system? Because I would expect it's very difficult. Like each country will have a different method of reporting extreme weather event, but also different ways of reporting what is an outbreak. And yeah, I was wondering if there's some kind of discrepancy that is the reason behind not including these countries, or how do you integrate this different, I guess, sources of data?
Correct me if I've maybe misunderstood, but the data that I was using was existing peer-reviewed articles with no limit on geography or date. So I was really just looking at what was out there. And so why historically maybe DRC hasn't been written on as much in terms of the links between extreme climatic events and then outbreaks, I don't have a full answer to that, other than research that has been written on it, say, yeah, again, when looking at Ebola or any other infectious diseases happening, outbreaks of cholera, even in DRC, they might more so have looked at interventions that have worked or taken a look in terms of what has happened to the sanitation system, but then not linked that back to climatic information. And why they haven't done that, it might be that that's just not as easily available in country, or the skill of any of that information isn't so good when you try and downscale it to DRC. But yeah, in terms of why other researchers haven't focused as much in these certain hotspots areas, I don't have a great answer for.
Thank you very much.
Any other questions? Okay, so join me to thank all the speakers this morning for the talks and their efforts to work on this climate crisis. Thank you.